Thanks. So some of the challenges of breeding perennial grasses and other forages such as alfalfa are that they have a self-incompatible mating system, which means that every plant, seed, and pollen grain is genetically unique. This really isn't any different than trees or animals. This does mean that the breeding is usually based on the selection of individual plants from space-planted nurseries, which you've seen some examples of that already this morning, but this is not the way the plants are grown by the farms. So for some traits such as yield, the breeding value of individual plants are sometimes evaluated based on the performance of their half-sib progeny in densely planted plots. The problem with this is that it takes a lot of time and it's difficult to do that. And we often try to avoid that. Another difficulty is that the pollen parents of the half-sib families are unknown, although in some cases you can determine that, but it takes a lot of work. The pollen parents are usually unknown and they're highly variable. And this diminishes our ability to detect genetic differences between the mother plants that we're selecting on. So following the 2017 Kernza conference in St. Paul, I acquired 29 lines, 14 lines from the Land Institute, lines from the University of Minnesota, and 10 lines from the University of Manitoba, and established 77 plants from each of these 29 lines, total of 2,233 plants on half-acre centers as my cycle zero base population to initiate breeding and develop genomic selection models for seed yields and for seed yield based on half-sib progeny trials in densely seeded plots and other traits in the Western US. And one of the main rationales for starting this project was recognizing that Utah it's the second dry state in the country. And yeah, we're just trying to develop plant materials that will be adapted to the semi-arid regions of the Western US, recognizing that the best plant materials in one location may not be the best plant materials in another location. And like I said, the other special thing I'm trying to do here is develop a genomic selection model for grain yield of the half-sib families in densely seeded plots, which is illustrated here in this figure. So in the first step of the process, I planted the mother plants on half centers in 2018 and harvested seeds in 2019 and 2020. Seeds from 2019 were planted in half-sib in plots, seeded into plots in the spring of 2020. Then we started harvesting those plots in 2021, and we'll harvest them for at least a couple more years. And then the idea is that based on that data, we'll go back to the mother plant nursery, the space planted mother plants, and select the best ones based on the performance of their progeny in those plots. But you can see that this process, this one cycle of selection then would take at least 5 or 6 years, and perhaps even longer if we really want to get at these persistent traits, this problem of persistence. So the idea is that we want to capture this in a model of genomic selection so that we can accelerate the selection based on this. So this just shows the mother plants probably from later in the summer in 2018. They're on half-meter centers, half-meter spacing within rows, 1 meter spacing between rows at this farm. This picture shows the harvesting of the mother plants in 2018. They're spaced on half-meter centers. We use rice knives to harvest all of the seeds from each plant, measure the seed yield, the total seed yield on each plant, and measure a lot of other traits. And then we hand selected 2 replicated sets of 100 seeds from each of 870 of the plants. I wanted to do more, but that was just as many as we could possibly do. This is an aerial image of 244, the 1,800 plots from those just before we harvested them in 2021, last year. Each plot is 3 meters long with 36-inch space between the rows. This 36-inch spacing is the same spacing that we use in our foundation seed fields. NRCS recommends spacing of 24 to 36 inches for seed production of a lot of the grasses, sometimes even more for rhizomatous grasses. Like I said, this 36-inch spacing is what we do in our seed production. We've had some fields, we have fields of crested wheatgrass now that are still in production after 16 years. My co-worker Alex Hernandez is using these aerial images to classify and determine the amount of grasses, the amount of intermediate wheatgrass in each plot, the amount of bare soil in each plot, and also the amount of weeds in each plot. So we can use this data to refine our yield measurements. The idea was the plots were supposed to look like this. And yeah, I mean, so obviously in the seed production, this would be, we're using the same row spacing as the seed production, but in our seed production field, we won't have these gaps within the rows. We have gaps so we can know where the plots are. I was hoping they'd look like this at the top here, but we were using a 2-row planter, and one of the planters when we were planting had some kind of problem and was dropping all of the seeds in a very short space, and we ended up with large gaps in every other row until I figured out that I had a problem and fixed it. But anyways, this picture shows during the first harvest of the half-sip plots taken on August 4th last year. Using a small plot combine to harvest each one individually. I stood by the combine with the bag, kept capturing the seed, so I got a feel literally for the weight of seed from every plot. It was kind of interesting. There was a lot of variation. These tables rank trait differences among the 29 germplasm sources in the training population. So here on the left, the 29 germplasm sources are ranked based on seed mass. And each line here would be the average of 77 plants, mother plants, evaluated as single space plants. The lines from the Land Institute show relatively high seed mass and low shattering and a lot of other important traits, standing traits for that. It was a bit surprising. The highest ranking lines for seed yield in the plots, I don't know what I'm doing there, were from Manitoba, at least in the first year of measurements, when there was relatively low competition between plants in the plots. And it's kind of interesting in the, what I'm doing. And then in the middle is the seed yield, the rankings of the lines based on seed yield per plant, space plants. And there you saw a mixture of lines from Manitoba and the land. This table is a trait correlation heat map of plot yield from the half-sieve families against 42 single plant traits. And so on the left side there, the strongest correlations were for the yield in the densely seeded plot came from the seed yield per plant of the mother plants measured in 2020 and 2019. Not too surprising. It was surprising that some of the other high trait correlations, like shattering, some of the highest yielding families in the plots were also had high shattering and low fruity threshing. And I think that is really kind of an artifact of population structure. Manitoba effect. The Manitoba lines were showing high yield, but they have poor shattering and free threshing traits. Some other interesting things, working down that list of correlations, there was correlations of tiller number based on single plant evaluations for tiller number of seeds per head, seeds per spikelet. Those traits were predictive from plant to plots. The seed yield per spike, not so much. In seed size was actually negatively correlated, but again, that could be an artifact of population structure. I think if we really want to look at the genetic trait correlations, we probably need to run these correlations within the 3 different germplasm sources. Like just look at run the correlations based just on lines from the Land Institute or just on lines from Manitoba, so we get rid of that effective population structure. So we have developed a genomic prediction model for yield of the half-sib families in densely seeded plots. There's a correlation 0.8 from our genomic predictions to the observed values. So it looks like we have a good genomic selection model that can be used to accelerate the selection. I would say qualify that and say I'm not quite certain we've got the final model. We need to look at persistence over years, but eventually we're going to capture all of this in a genomic selection model that can be for rapid selection, like what Lee and Praveen are doing. We've also detected significant QTL effects or gene effects on 9 of the 21 wheatgrass chromosomes. Jared pointed out earlier, we know that there's probably hundreds or maybe even thousands of genes here that are affecting yield, but we are detecting some of the larger, when I say larger effects, we're still really only talking about maybe 1 or 2% of the phenotypic variance. But and then finally, I was going to mention here the heritabilities on the half-sib plots were comparable to heritabilities of single plant yields. If we analyze the data, if we treat the data the same way, we get about the same heritability values, but in actuality, the heritabilities of the half-sib plots are probably twice as good because we're not controlling for the effect of the pollen parents. So in other words, with those, the heritability for based on yield on half-sib plots is actually probably quite good. In the sense that what we would gain from selection would be quite a bit better than basing it on single plant yield. Let's see, I'm going to skip over this slide. Try to do this quickly. We've organized with funding from this FFAR grant. We've been organizing these small plot yield evaluations. We have 40 half-sib families from Minnesota, 40 from the Land Institute, 20 from my program, planted in replicated trials, 2 locations in Minnesota, 2 locations in Kansas, and 2 locations in Utah. This is an aerial image of the irrigated experiment in Logan. It's basically the same design, that I'm using in my training population. Interesting thing here is that there's a weak but significant correlation between the 100 lines in the FFAR trials between one of the experiments in Kansas and the irrigated one in Utah, the 0.29, but there was no correlation between genomic prediction of yield from my training population to either of these two FFAR trials. So this probably indicates significant genotype by environment interactions, but it could also be the lack of correlation in the genomic prediction model could be an effect of a lot of the lines. It might be just difficult to extrapolate from the germplasm in my training population to the more advanced lines that are in this FFAR trial. So there's several things probably going on here. So in summary, we're starting to see many significant trait correlations between single space plants and yield from densely seeded plots, at least in the first harvest year where competition is still minimal. We need to monitor yield in the plot as the plots fill in and become more competitive over the next few years. We have what looks like a good genomic prediction model, but there does appear to be limitations. We may not be able to extrapolate that to more advanced germplasm. Yeah, and I'd also like to mention we're using, the multispectral sensors and looking at using multispectral sensors, ground penetrating radar, and other techniques to measure and understand yield, ground cover, and maybe even root development in these yield trials. Thank you.
So maybe I didn't understand very well, but there was a very low correlation between the yields in the space plants and the yields in the pots, or did I miss something, or was a different thing?
So the question was quick clarification for the yield on space plants versus yield on plots. Of the many traits that I measured on the single space plants, the trait that had the best prediction or the best correlation with yield in the plots was the yield per plant. But it was 0.58. So it's not we'd like that to be stronger, but there is a significant correlation there. I think with that said, I think we really need to wait a couple of years and look at these plots over a few more years, because I think those what I'm calling densely seeded plots, yeah, there's 100 plants in maybe a 2-meter space, but it's still relatively low competition compared to what it's going to be like in a couple of years. So with that low competition, it's kind of, the issue with single space plants is there's no competition. Okay. And then the question is, do I have enough, was there enough competition in my yield plots? Maybe not. Maybe that correlation between yield in the plots and yield in the plants will go down as those plots become more active. I don't know if that answers the question.
What does that mean for the breeding programs?
Question is, what does it mean for breeding programs?
Well, I think what it means is, what we'd like to do is try to, we want to be able to breed for yield the way that the farmers grow the plants. And I think we've been doing that. Lee's been doing, Minnesota's been doing that, but I think as we move further along in the process, we'd like to sort of refine that and really zero in on, you know, refining those yields and getting the maximum yield the way that the farmers are growing it.